Hello everyone, welcome back. I will be continuing the numerical question series for ICAR JRF exam plant sciences and SRF exam for genetics and plant breeding and seed science and technology, and these are also useful for all agricultural MSc entrance exams. So this is the part 4 of numerical question series, so where I will be mainly dealing with the plant breeding questions. Along with the plant breeding, I have few questions from the genetics which I received from some student. So I'll be discussing those questions first and then shifting to plant breeding. And this will be the last video for numerical questions, after which I have two more videos in a row, where in one video I will be covering the most important topics to be covered in seed science, like how you can cover the seed science in a day, and secondly I will be covering the experimental designs in plant breeding like RBD, CRD, LST. So I will be giving a overview of what are these experimental designs, like where they are used and what type of questions you can expect in the JRF exams. So it would be very short and very crisp, so make sure to attend or make sure to watch this video once it is uploaded. So for all the further updates, please subscribe our channel and also join our WhatsApp and Telegram group, so that once we upload the video you'll get the notification. Okay, let's get started. We'll start with the first question. This is regarding the chromosome mapping. Okay, so the question is very simple. It says like the map distance between A and B gene is 3 units and B and C is 10 units and C and A is 7 units. So they are asking for order of genes in the linkage map constructed. So they are just asking to construct a linkage map based on the given data. So this is simple mathematical calculation, where if it is 10 which is higher than other two, it will be definitely 7 plus 3. So C to A, and before that we can also construct, or after that it is A to B. So if A to B is 3 units and C to A 7 units, obviously B to C will be 10 units. So this is how you can just make out the order. The order of gene in the linkage map will be either CAB or BAC. So we cannot exactly tell based on the recombination frequency the order of genes, like whether it is BAC from the centromere or CAB from the centromere. So for that we have to do gene sequencing. Only based on the recombination frequency we can just tell whether this gene A is located in between or which one is located in the middle of the other two. So that is what we can say based on this linkage map, whether it is ABC or BAC or anything. So the order is CAB, that is B is in, like A is in the middle and C and B are at the extreme end. Either it could be CAB or BAC. I hope this is clear. So this is a simple mathematical calculation. Okay. Next question. Assume a population in R.D. Weinberg's equilibrium with this genotypic frequency, where capital A is 0.25, capital A small a is 0.5, small a is 0.25. So if you remove all the homozygous dominants and allow the remaining population to reproduce, so again under R.D. Weinberg conditions, what will be the frequency of homozygous dominants in the next generation? So what they are saying, like capital A is 0.25, which means P is equal to 0.5 and capital A small a is 0.5, which is 2pq equal to 0.5. Next it is small a, which is 0.25 again, where q is equal to 0.5. So they are saying out of this they are removing this complete population of dominant homozygous. So what will be the rest that are there in the population? It is 0.75. The 0.75 of the frequency of population will be there in the population. So the rest 0.25 is removed from that. So now we have to calculate the new frequency of q here. So new frequency of q will be, we know that q frequency is 0.5. So but what about the total population? The total population is 0.75. So 0.5 divided by 0.75, what will be the value? It will be 0.66. So q value will be 0.66. So if q is 0.66, what will be the p? P is 1 minus q, so it would be 0.34. So then what about p square? So they are asking about frequency of homozygous dominant in the next generation. The p square will be somewhere around 0.11. So your answer will be 0.11. I hope this is clear, right? So we have to just cancel this 0.25 if it is cancelled, or if it is removed. You have to just add the other two frequencies. It is 0.75, which is the total frequency of population that is present in the next generation. So we already have this q frequency, which is not changing because none of them are removed from the population. So 0.5 by 0.75 will give you the frequency of q in the next generation. So from the frequency of q in the next generation, we can make out the frequency of p, which will be simply 1 minus q, where it is 0.34. So p square is again 0.34 into 0.34, which would be 0.11. Next one, a female Drosophila heterozygous for tragene, capital TRA, small TRA, is mated with a male homozygous for tragene. So which of the following will be the proportion of female and male progenies? So they are just saying that heterozygous female is crossed with homozygous male. So what will be the proportion of female and male? This is what they are asking. So now we have to cross female with male. So XX with XY. So for that we have to know that tragene is autosomal gene. This is not a sex-linked gene, this is the autosomal gene. So how do we denote this autosomal gene along with the sex chromosomes while doing the cross? If it is a sex-linked gene, we will be just denoting it as XTRA with XTRA. So this is how we do it, right? But since it is autosomal gene, we cannot do like this. And also if we just separately cross capital TRA small TRA with small TRA, we will get different combination. That is perfectly fine. But how do we find the male and females in it? Like how do we know that which one is female and which one is female? So to know like which one is male and which one is female, we have to cross in such a way that both should be there while doing the cross combination. That means both the chromosomal genes like sex chromosomes along with the autosomal genes, both should be present. So for simplification, I will just mention it as capital TRA, small TRA in the above. I'm not using the superscript, but I just mentioned it in the above. So crossed with male, which is small TRA and small TRA. So this is how we can do it, right? So now if we just look into the combinations, so X capital TRA, X from male, small TRA, first combination. Second one, small TRA with again small TRA from male. Third, X TRA, capital TRA from female, Y from male. Next, this is capital TRA. So small TRA, Y, small TRA from male. So out of this four combination, what will be the sex of these individuals? The first one, obviously it is XX, it will be female. So secondly, it is XX with both homozygous tragene. I will just explain this later. Okay. Third one, X and Y, it is male, and fourth, it is male. So what will be the sex of this individual with homozygous for tragene? Okay. For that we have to understand the function of this tragene. So the tragene in its recessive homozygous condition, just listen carefully. In its recessive homozygous condition, which is the autosomal gene, this will transform the females into males. So this will transform female into males. This particular gene, which if it is present in the autosomes, it will transfer the female into males even though it has XX sex chromosome. Even though sex chromosome is X and X, still it will convert the females into males, and it doesn't have any other function in the males, both in males and females. Only it will transform females into males if it is present in the female, only in the homozygous recessive condition. So you can clearly observe the homozygous condition is also present in the male, but it doesn't have any function there, so it will be obviously male only. So here, since it is in the homozygous recessive condition and it is in the female, it will get transformed and the progeny which is formed will be male. So the ratio becomes 1:3, that is 25% will be male and 75% will be female. 25% will be female and 75% will be male. So this will be your correct answer. So in order to solve this question, you have to know the function of this tragene, which is unique here. Okay, just try to remember, one thing is it is autosomal, secondly it is recessive homozygous, will transform female to male with no other function. So you have to remember these 2 points. Next one. So if the number of segregating loci is 10, the frequency of the plants fixed in the F2 for all the positive alleles will be which of the following? They are asking for positive alleles. Positive alleles means if it is capital A and small a, capital A will be the positive allele. So this is for only single locus. In the single locus, in the F2, if we are just crossing the F1, if it is F1, let us take an example. So we will have 4 different combinations, right? Out of this, what will be the frequency of the probability of getting capital A? It will be 1 by 4. So this is half, 1 by 2, and this is again 1 by 4. So this is for a single gene. So now we have to calculate for 10 genes. So it is simply 1 by 4 in whole to the power of 10. So this will give us the total number of positive alleles if there are 10 genes. Okay, so the answer would be 1 by 4 to the rise to the power of 10. Next one. From the marriage between two heterozygotes for albinism, what is the chance that first would be non-albino? For that, we'll just do the cross. We'll just take it as small a as a albino. Albinism is caused by a recessive allele. So albinism is usually caused by autosomal recessive allele. So that we have to know, this is not sex linked, it is autosomal recessive allele. So in this case, if we are just crossing two heterozygotes, we already know like the combination, right? Like the resultant combination will be capital A, small a, and small a, small a. So this will be 1 by 4, and this is 1 by 2 again, and this is 1 by 4. So out of this, so 1 by 4 is albino and 3 by 4 is non-albino. So always when there is a child which is born, so frequency of getting the non-albino or probability of getting non-albino will be always equal to 3 by 4. Whether it is first child, second child, third child, or nth child, the probability will be always equal to 3 by 4 for non-albino, and for albino it will be 1 by 4. So that frequency won't change if they are if it is between true heterozygotes. Clear? Okay, next one. So in this case, we'll be starting with plant breeding now. So in this case of dihybrid cross, how many types of gametes will be produced by F1? So this is again same as the Mendel problems that we solved in the genetics. The difference is they are mentioned about dihybrid cross. So what is dihybrid cross? It has two genes which is resulting for two different characters, or which is governing two different traits. For example, A is governing some trait and other gene B is governing some other trait, might be color and might be plant height or anything else. So in this case, they are just asking like how many different types of gametes are produced. We know the general formula, it is 2 power n. So n is number of segregating genes. So here it is 2 power 2. So it will be 4. So your answer should be 4. So don't get confused just by seeing the dihybrid cross. So dihybrid simply means that it has two genes. That's it. And also don't confuse this with quantitative traits. This is not quantitative, again this is qualitative, because two genes are governing two different traits here, not a single trait. Next one. The number of all possible crosses excluding the reciprocal crosses in the diallele cross is calculated by which of the following formula? So diallele cross is simply a population improvement program in which we can predict the crosses. So if they are saying that to exclude the reciprocals, it will be n into n minus 1 by 2. If they are asking for excluding reciprocals. Reciprocals means reciprocal crosses. If we are using like capital A as female in first case, cross with small a as male, so we can just reciprocate and again do the cross, right? So this is how it is. This is just a simple example I am giving. So if they are asking for exclude these reciprocals, this will be like n into n minus 1 by 2. If they are asking like including reciprocals, it will be just n into n minus 1. Just remember these two formulas. It can be very much important, like it can be directly asked in the exam. So excluding reciprocals, it will be just half of it. Like including reciprocals means total number of combinations. If total number of combination is n into n minus 1, if they are asking to exclude reciprocals, it simply means that to cancel 50% of the total combination. So to cancel the 50% of combination, you just need to divide it by 2. So that is how you can just remember excluding reciprocals and including reciprocals. Next one, the effect of self-fertilization for n generation on the frequency of one of the two homozygotes, that is A and small a or small a at a single locus can be calculated by using the formula. They are asking for single locus, that means gene is 1. The generations are n. The simple and direct formula for this is 2n minus 1 divided by 2n plus 1. So this will be the simple formula for calculating the frequency of one of the two homozygotes. So since they are asked for only one gene, like this is the simple formula where n is equal to number of generations. If they are asking for not for multiple locus or more number of genes, either it could be 4, 5, 10 or any number, so here it will be having another letter which is denoted as m. M is number of genes. So this is number of generations and m will be number of genes. So if just make sure to see whether they are asking for a single locus or multiple genes. If the number of genes are more, you have to add the superscript m here, like it should be squared or it should be in the bracket, it should be m, where m is number of genes. If it is like, since it is 1, like we can directly write it as 2n minus 1 divided by 2n plus 1, sorry 2n plus 1 if it is a single gene. But if it is more than one gene, we have to denote the number of genes here at the outer bracket. If it is 2, we have to mention it 2, and this is how we can calculate the frequency of the homozygotes, like one of the two homozygotes. I hope this is clear. Just remember this general formula and don't even forget about this one, like to add the m in the outer bracket if they are asking for more than one gene. Okay, and this m and n, it might be interchanged in the some of the books, so this is not fixed. M can be mentioned as n can be mentioned as m, but in the inside the bracket, n always refers to number of generations. That you should be make sure that you know that. And outside the bracket, whatever the like an annotation is provided, it just simply means that it is number of genes. Okay, inside the bracket it is number of generation, outside the bracket it is number of genes. Next one, the similar one, the effect of selfing for n generation on the frequency of both homozygotes with respect to single locus can be calculated by which of the following formula. So here the formula is 2n minus 1 divided by 2 power n. So this will be the simple formula if they are asking for frequency of both homozygotes. The earlier one was one of the homozygotes. If they are asking for both the homozygotes, it is 2 power n minus 1 divided by 2 power n. Again, n is number of generations. Next one, the effect of selfing for n generations on the frequency of heterozygotes. Here they are asking for heterozygotes with respect to again single locus will be which of the following. So the formula for this is 2 by 2 power n plus 1. So this can be also written as 2 power n into 2 power 1, which is 1 by 2 power n. So in the option, either you will find 2 by 2 power n plus 1 or 1 by 2 power n. So don't get confused. Even if it is given in the form of 2 by 2 power n minus n plus 1, you have to tick it, or if it is given in the 1 by 2 power n, you should mark it as a correct answer. That would be the correct formula. Next, so in which of the following 4 gene arrangements the greatest number of crossovers will be needed to produce capital A, capital B, capital C, capital D and small a, small b, small c, small d. So this I have also explained in the previous video. So I will be just expecting the answer from your end. Just kindly comment on this video. Okay. So if the performance of the two parents in the hybrid is 10 and 6 units, what will be the following? Which of the following is the case of heterosis here? So this is what they are asking. So heterosis can be of positive and negative. So any value outside the 6 and 10 will be the heterosis. right? Like it's just superiority or inferiority about f1, inferiority or superiority of f1 over their parents, right? So any units, any value outside this 10 and 6, between 10 to 6 or 6 to 10 will be the heterosis. So which are the values? It is only 4 and 12. So all other two options, other three options are between 6 and 10. So the answer will be 1 and 2. So this will be the case of heterosis. Next one. Two inbreed lines of the beans were intercrossed. The F1 the variance of the bean length is 2, was 2. The F1 was self to obtain F2 and the variance of the bean length is in the F2 is 7. What is the broad sense heritability of bean length in the F2 population? So broad sense heritability is equal to variance, genotypic variance by phenotypic variance, right? So based on the given data, it's very easy to understand that variance environmental or variance due to environment is just 2, since if variance that we see in the f1 is purely because of the environment, so directly we can write it as ve is 2, and the total variance that we are getting in the f2 is total variance in the f2 is 7. So out of this total variance is nothing but phenotypic variance. So phenotypic variance is genotypic variance plus variance due to environment. So now we have to find the genotypic variance, right? So we know the environment variance and total variance. It will be 7 minus 2, which will be 5. So 5 divided by 7 will give you the broad sense heritability. So just comment your answer on this video. It is just multiplied with the 100, which will give you the value or the value of broad sense heritability. Okay, so answer will be 0.71. Okay, next one. The possible number of double cross hybrids when the number of inbreed is 10. So what is there is a direct formula to find out the number of double crossover. So it is n into n minus 1, n minus 2, n minus 3 divided by 8. So this is the direct formula to find out the possible number of double crossovers in the hybrids when they have given the inbreed line. So just calculate and just comment your answer. So next one, for 12 genes affecting the quantitative trait, there will be dash number of different phenotypic classes. I already mentioned the formula for this. It is just 2n plus 1, where it is 12 into 2 plus 1, it is 23. So there will be 23 different phenotypic classes. Next one, what is the smallest perfect F2 population when 4 genes are segregating? This I have also mentioned this in the one of the previous video. The direct formula to find out the smallest perfect population is 4 power n, where n is number of genes segregating. So it is 4 power 4, which is equal to 256. So 256 is your answer. Next one, calculate the performance of synthetic variety in the second generation developed by using 10 inbreds when the mean performance of synthetic in the first generation is 100 units and the average performance of the inbreds or the parents is 50 units. So again, to find out the synthetic variety, like performance of synthetic variety in second generation, has a direct formula where it is synthetic 1 in the first performance of a synthetic variety in first generation minus synthetic variety in first generation, again minus the mean performance of the parent divided by n. N is number of inbreds. So this is the direct formula. So if you just substitute, so it is 100 minus 50 divided by 10. So the answer would be 95. Next one. Calculate the inbreeding depression when two parents are crossed to produce F1 with the mean value of F1 is 30 units and F2 is 15 units. So they have given the value of F1 as 30 units and F2 as 50 units. Now we have to find the inbreeding depression. So to find out the inbreeding depression, it's simply F1 minus F2 divided by F1. So that is 30 minus 15 divided by 30, which is equal to 1 by 2, which is 0.5. Next one, the possible number of three-way cross hybrids when the number of inbreed is 8. So earlier I told you about the double cross hybrids, which is n into n minus 1, n minus 2, n minus 3 divided by 8. So for three-way cross, it's simply n into n minus 1, n minus 2 divided by 2. So for single cross, it is just n into n minus 1 divided by 2. So possible number of single crosses, three-way crosses, and double crosses. So this is how we can find it. So if you just substitute, it is n into like 8 into 7 into 6 divided by 2. So the answer will be 168. So these are the questions that I wanted to discuss in this video. So this will be the last video for last video of this numerical question series for ICAR JRF Plan Science exams. If you still have any questions, you can directly post it in our WhatsApp or Telegram group, or either you can also directly send me the questions in the WhatsApp. So I'll be just taking the questions and answering in personal. Okay, so anything else, if you have any other doubts, if you want any other topics to be discussed, so just comment on this video. We'll definitely take that and prioritize that, and we'll just try to do it. Okay, so if you're liking the content, please like, comment, share, and subscribe our channel and encourage us. Thank you.